Hello and welcome to the Gene and Cell Engineering Virtual Summit. Today's summit is our inaugural summit presented by GenScript. And before we get started, I would like to draw your attention to a couple of housekeeping items. So all talks will be available on demand following the summit on LabRoots in the auditorium for the next 12 months. You can use the chat box to send questions during the Q&A after each talk. The exhibit hall and booths are open. You can visit during the break. And for event support or help, please click the help desk on the bottom of the page, and you may also send a message to us. If you're interested in career opportunities by GenScript, you can find that as well in the in the lobby. So please don't forget to take our customer feedback survey as well, and you can share about this conference on social media using the hashtag GCE Summit. Our opening remarks will be provided by our CFO, Xinyu Wei, and following that, there will be our opening keynote by Alexander Marson, Director at the Gladstone UCSF Institute of Genomic Immunology. Please take a look at the schedule of events. Track 1 will be concurrent to tracks 2 and 3. And right at the end, our closing keynote will be provided by Joseph Mellenhorst, professor at the University of Pennsylvania, and the closing remarks by our very own Ray Chen. To help you navigate the event, I'd like to draw your attention to the lobby. You will be able to see a welcome video on the top left. It would be great if you could fill out our feedback survey. You can use the chat box to communicate with the other attendees. And of course, once again, if you're interested in career opportunities at GenScript, there is a link to that there, and as well as the event support. Here's what the auditorium looks like. Today's talks will all be recorded. You will find all the talks here in this auditorium page, available on demand for the next 12 months, starting from today. And they will be made available immediately after the summit. So you can always come back to the auditorium. You can always go back to the auditorium for the next talk. This here is the exhibit hall. On this page, you will find 5 booths for different services, and at each booth, you will find supporting files and ongoing promotions. You can send us a message here if you're interested in a particular service. Before we continue, I'd like to acknowledge the GenScript team who have helped us put together today's summit. And please take a minute to read through the careers blurb in case you're interested in applying at an opportunity at GenScript. Now I will hand over to Xinyu Wei for the opening remarks. Thank you so much. I look forward to seeing you soon.
Hello, everyone. I'm Xinyu Wei, Chief Financial Officer of GenScript Biotech. It is my great honor to welcome you to GenScript's first Gene and Cell Engineering Virtual Summit. We're excited about the program that we're about to share with you today, and thank you to the distinguished scientists who have generously agreed to participate. Over the past decade, we have all witnessed significant advances in gene and cell engineering. Today's speakers are leading and shaping the present and future of genome editing applications and technologies. For example, the development and continued improvement to the CRISPR-Cas toolkit have transformed genome editing, facilitating development of preclinical models of diseases and enabling treatment at the clinic that were unimaginable a decade ago. During track 1, titled Expanding CRISPR Toolbox, you will hear from Dr. Kleinstiver's and Dr. Zurith's work about engineering Cas variants for improved genome targeting and editing efficiency. Advances in cellular engineering, as exemplified by T-cell engineering, have enabled the development of powerful new therapeutics for cancer treatment, offering hopes to millions of patients worldwide. Today, 2 keynote speakers, Dr. Marson and Dr. Mellenhorst, will discuss progress on the application of CRISPR tool in developing T-cell therapeutics and elucidating determinants of CAR T-cell responses in chronic lymphocytic leukemia. The GenScript family is proud to be part of this wonderful journey and honored to have endeavored to make research easy for so many scientists worldwide by providing superior products and services. Since our foundation in 2002, it has always been GenScript's mission to make people and nature healthier through biotechnology. Therefore, GenScript is in pace to facilitate gene and cell engineering research and its leading role in cell therapy. GenScript's original business unit, the Life Science Group, has harnessed the power of synthetic biology through its innovative gene synthesis platform, providing critical support to investigators worldwide developing gene and cell therapy strategies, from oligos to complex gene synthesis, plasmid assembly for clinical relevant viral vectors, and precision mutation libraries, which expedites the viral vector optimization. These services are making a difference for thousands of investigators in this field. Our internally incubated biologics CDMO business, GenScript ProBio, works closely with global partners and strives to become a leading gene and cell therapy provider. Not to mention Legend Biotech, a global cell therapy company with cutting-edge platforms, robust pipeline, and potential best-in-class products to bring miracles to millions. GenScript's fast-paced progress has only been possible through key partnerships, as exemplified by established agreements with Dr. Zhang Feng's group at Broad Institute of MIT and Harvard, and more recently with ERS Genomics. These partnerships have allowed GenScript to provide researchers worldwide access to a high-quality CRISPR portfolio, including synthetic single-guide RNA, guide RNA plasmids and libraries, and cell line engineering services. Throughout today's presentations, you may find out how GenScript's products and services have facilitated our speakers' research. We invite you to take this opportunity to reach out to our knowledgeable product managers, who will be available at several booths focused on different services. Find out GenScript can make your research easy. Lastly, I want once again extend my deepest gratitude to all the scientists speaking today. To our audience, thank you for attending today. I hope that this event sparks great ideas and partnerships to help achieve your research goals. Thank you.
Good morning. I think you're hearing me now. Well, first of all, it's an honor to have a chance to present our work today, and thank you for that opportunity. I'm excited to have a chance to tell you a little bit about the work that we're doing in my lab and at the new Gladstone Institute of Genomic Immunology. The work in my lab is really focused on trying to understand DNA programs, gene programs that control the behavior of cells in the immune system, particularly human T cells. And more, we're using gene engineering to perturb these programs and really understand their function, but also thinking about how we can actually rewrite these DNA programs in human T cells to transform the actual cells into cellular therapies to treat a wide range of human diseases. So before I go on, I want to show you some disclosures that I'm obligated to show. I do want to also highlight that we've had a collaborative relationship with GenScript, including discounts on reagents, and my presentation today is part of that collaborative relationship. So we, like many of you, have been inspired by the success that we're already seeing in human patients with genetically engineered cell therapies. The currently FDA-approved cell therapies, are the CAR T cell therapies, are where a patient with certain types of cancer have their T cells removed from circulation. The cells are then genetically engineered outside of the body. Currently, the approved products are engineered with viral vectors, either lentivirus or retrovirus, that can insert transgenes for chimeric antigen receptors, or these artificial receptors that will redirect the specificity of those T cells. The current vectors insert the genes for these CARs, or chimeric antigen receptors, into non-targeted sites in the genome. But that's sufficient. That genetic material goes in somewhere in the genome, and that's sufficient to actually transform the specificity of those T cells, which can then be reinfused back into the patient. And the results have been really astounding for many patients with certain types of hematological malignancies, blood cancers. And I'm showing there's now a number of FDA-approved CAR T cell products that are available. But we think that this is really just the tip of the iceberg of what's possible. In particular, we'd like to see more robust responses for these hematological malignancies. We'd like to expand the opportunity to use genetically engineered cell therapies, not just for blood cancers, but also for solid tumors, where they've had limited success. And beyond that, we'd like to make these treatments more accessible, more potent, and we're even thinking about how do we expand beyond cancer? How do we start engineering cells for autoimmune diseases and infectious diseases and other classes of human disease? And we're inspired to think that all this will be possible because, along with the advances in cellular immunotherapies, there's also been remarkable, really revolutionary advances in the power to edit the genome at precise places. And in particular, all of us are inspired by the power of Crispr to do targeted gene engineering. So we have put a lot of work into being able to perform a number of molecular edits at precise places in the genome, including removing genes that are limiting the function of immune cells, fixing individual mutations, and what's been probably technically the most challenging, but where there's probably the most therapeutic opportunity, is the ability to take a long stretch of DNA and either add it to a targeted site in the genome or rewrite a targeted site of the genome to really start reprogramming the functional properties of that cell and making it behave how we want to transform it into an optimal therapeutic. So, so this one slide summarizes many years of work in the lab. We were watching the power of CRISPR in cell lines and animal models as it was first rolled out. And we really wanted to develop tools to use it in primary human immune cells. And it turned out that there was a delivery challenge. We made an initial headway through an amazing collaboration with Jennifer Doudna, one of the pioneers of CRISPR. where we started using recombinant Cas9 protein that we could load in vitro with a guide RNA to make a ribonucleoprotein or RNP. And we found that these RNPs could be electroporated into human immune cells, human T cells, and that was sufficient to get very high delivery of these RNPs and drive high efficiency gene ablation. We also found that we could start co-electroporating an extra piece of DNA that had homology arms to either side of the cut site, and that extra piece of DNA could trick the cell's homology-directed repair machinery into using the sequences from that template to repair the cut site. And that allowed us to actually start rewriting with specific nucleotides at a specific site in the genome. Just summarizing many years of work, we've now increased the efficiency, we've increased the size of these payloads, and I'll talk a bit more about that today. And we've started to expand this beyond just T cells to other cells in the immune system, including B cells and NK cells and CD34 cells, which should include the hematopoietic stem and progenitor cells. So we think this is a very flexible way to do targeted rewriting of the genome in cells that can be cultured and expanded ex vivo, and that this allows us to actually start probing the genome and start actually adapting this for clinical use. And we're working very actively towards clinical trials with this non-viral CRISPR-based ability to rewrite cellular therapies. So for any cellular therapy, one of the most important things that we can do for a T cell is direct what it will recognize, direct its antigen specificity. And there's been a number of ways to direct antigen specificity. I mentioned already these chimeric antigen receptors, or CARs. Michelle Sadelain very beautifully showed that there's a benefit of putting the chimeric antigen receptor into the cell's endogenous T cell receptor gene, and that by doing that you can actually control the levels at which the chimeric antigen receptor is expressed, and that there's a benefit for the function and durability of those cells. We've also put a lot of work into being able to take the cell's own T cell receptor and actually rewrite the TCR alpha and beta pair that determines its specificity. So with either of these tools, we now are trying to have the opportunity to develop a totally non-viral, flexible, CRISPR-based system to introduce these receptors. So a few years ago, we were really seeing what we could accomplish, and as we started to improve our ability to do this completely non-viral rewriting of endogenous DNA sequences. And we wanted to see, could we actually rewrite the cell's own T cell receptor, as I mentioned. Now, as I said, the T cell receptor has two components, a TCR alpha and a TCR beta. So we wanted to see, could we simultaneously replace both? And this was through a collaboration with Tony Rebus's lab at UCLA. But we wanted to see, could we actually put in a huge piece of DNA, 1,500 base pairs, targeted into the cell's TCR alpha. But we'd actually knock in a polycistronic DNA sequence that encodes the full-length TCR beta, and we just put the variable part of the TCR alpha in, and then fuse it in frame with the rest of the cell's own TCR alpha constant region. If this is successful, it should put in both the beta and the alpha with no extra gene regulatory elements. This is driven entirely off the cell's own endogenous TCR alpha gene regulatory elements, so the expression should be tightly regulated. We started this by seeing, could we put in a T cell receptor that's been well studied and recognized as the cancer antigen NY-ESO. And the results got us really excited. You can see the control cell on the y-axis. We're staining for cells that recognize this NY-ESO antigen. You can see the control cells don't recognize this, but we've started to achieve high efficiency knock-in, where we can actually make these engineered cells that recognize this cancer antigen. And we published this a few years ago, so I'm not going to go through all the details, but I'll just say we put these cells through a whole battery of tests. And in vitro, these non-virally engineered cells will recognize cancer targets in a way that depends both on the antigen and on the right MHC background. They'll recognize them, they'll secrete proper cytokines, and they'll kill the cells in vitro. This also works in vivo. We can actually put human tumors into an immune-deficient mouse, and these non-virally engineered human T cells will home to the tumor bed in the mouse and actually control tumor volumes. So we think that this is a very powerful, flexible system to put big sequences in that rewrite antigen specificity. Now, in order to do this, we've also continued to work on how can we manufacture these cells better to get higher efficiency, higher yields, and march towards the way that we will actually engineer these for future cell therapies. So I'm going to tell you a little bit about that. And then in the second part of the talk, I'll talk about how we're also discovering genes that we can modify in these cells to enhance their functional properties and make them into more potent cellular immunotherapies for cancer. So what are we doing to actually improve the gene engineering? I guess early last year, we published a paper that was led by David Wynn and Theo Roth in my lab, where we wondered if we could actually increase the efficiency by making sequence modifications to this homology-directed repair, this DNA template that we're using for gene knock-in. And the idea was, well, Cas9 binds to DNA. Could we actually trick the Cas9 into binding to the template? And we know that we can actually engineer Cas9 with nuclear localization signals. Could these nuclear localization signals actually pull this DNA into the nucleus and increase the efficiency of delivery and repair? There might also be added protection through this through other mechanisms. For example, maybe putting these proteins on the ends of these templates would protect them from degradation. We don't fully know the mechanism, but we came up with a way to do this. The idea was that it's been known in the literature that if you have a Cas9 binding site that's truncated, Cas9 can bind to it but not cleave that DNA. So we put these truncated Cas9 binding sites onto either end of the DNA template. And these binding sites match up with the cut site that we're introducing in the cell's own genome. So now the same RNPs can bind to the template and cut the DNA. And we found that this was a tool that allowed us to start increasing the efficiency of knock-ins. But one thing that we've also noticed is that if we get a lot of DNA into these cells at higher concentrations, it starts to be toxic to the cells. Now, one thing we've speculated and seen some evidence of is that maybe single-stranded DNA is better tolerated by the cells. So what I'm going to tell you about in the next few minutes is an approach to see could we adapt this system where with these binding sites on either end of the template to a single-stranded DNA approach. So this is unpublished work that's been led by Brian Shai, who's an MD PhD in my lab. He's also the first clinical cell therapy fellow in the lab medicine department at UCSF. So has a strong interest in bringing these technologies towards actual clinical grade manufacturing. And he's been working very closely with a talented technician in the lab, Vivo. What they've come up with is an approach to adapt this DNA binding system, which we call the shuttle, to single-stranded DNA. Now, Cas9 requires binding to double-stranded DNA. So the trick that Brian has come up with is to actually put double-stranded DNA onto the ends of these. And we found that we can actually make a single-stranded DNA template with small oligos that are complementary to either end of the site, providing these truncated landing sequences, these truncated target sequences to Cas9 on either end of a single-stranded DNA template. And the results have really been eye-popping. What we find is that in contrast to the DNA, the double-stranded DNA with these binding sites, where we do see a benefit at the lower concentrations, but that sort of starts to fall off at higher concentrations of the template, we can actually load these cells with high concentrations of the single-stranded DNA templates with the binding sites, these Cas9 truncated sequences. We can actually get very high concentrations of the template, which gets to huge knock-in efficiencies. So we can actually knock in, for example, a CAR. Now, this is a big DNA construct. We're getting up to 40% knock-in into the TCR locus with very high efficiency. So just a little flavor of where we're going to take this manufacturing capability. I want to show you some of the actual products that we're making with this and trying to move these into clinical trials. There's now a anti-BCMA CAR that's FDA approved for multiple myeloma. We wanted to see if we could make a completely non-viral version of this that has the benefits of going into the TCR locus, which we know based on the CD19 work by Michelle Sadelain has a benefit. Can we do this non-virally, targeted, and hopefully start to do this in a way that brings down cost and increases accessibility? So we're getting very high efficiency knock-in with this system into the BCMA CAR. We are also collaborating with other groups. We're working closely with Phil Greenberg, Michael Wilson, and Fyodor Urnov to develop TCR T cells that recognize viral peptides. We're going after the JC virus, which causes a horrible neuroinfectious disease in certain immunocompromised individuals. And we're able to get a high knock-in efficiency of the Tcell receptor that recognizes a viral peptide. And we're also working with Hideo Okada and CERM and the Parker Institute for Cancer Immunotherapy, PICE, to make Tcell receptor T cells that recognize an antigen found in certain kinds of pediatric brain cancer. And we're able to manufacture this at high efficiency. So now we're at the stage of really thinking about how are we going to manufacture this in a way that's going to be compatible with the standards required for a clinical trial, or so-called GMP manufacturing. So we've been looking for sources for all the reagents for Cas9 and the guide RNA and all the things that are required to grow these cells. But now we have a new requirement for single-stranded DNA, long single-stranded DNA, that can be used in a clinical manufacturing process. And this is where we've benefited from a wonderful collaboration with GenScript. We actually turned to GenScript and asked, and GenScript is working towards a manufacturing capability that will be GMP compatible in the future. So we asked if they could actually start making these long single-stranded DNAs that encode the template for a car, for example, but this could be other things in the future. And what we found, to our real pleasure, and is that these templates are not only have a path towards being GMP manufacture capable, but actually showed a marked benefit over the single-stranded DNAs that we were generating internally. And we found that we could actually load them with even higher concentrations of these GenScript manufactured templates, and that actually continued to further increase the knock-in efficiencies, whereas even at the highest concentrations that we used, we started to see fall off with our home templates, suggesting maybe there's some impurity in the way that we're generating these. And that translates to more live cells, and most importantly, to a high number of live cells at the end that have successfully knocked in these templates. And that's what we want for a clinical efficiency, so that we can actually manufacture large numbers of cells that would be compatible with the cell therapy. So in our latest unpublished data, Brian and Viva have now actually done this in a way that is at a clinical scale. They've taken a leukopak from healthy donors and actually used an electroporation device. We used the MaxCyte device that would ultimately be compatible with GMP, with cytokine conditions that would be compatible with the way we'd manufacture these. And we've used the GenScript material to make BCMA CARs at scale. So you can see we're getting very high efficiency even at this large scale that would be used in an actual clinical cell therapy. And we find that these cells are able to generate really, over 1 billion cells within less than 10 days. Within 10 days, we're able to generate well over 1 billion cells, putting us well into the range of where we, and of CAR positive cells, putting us well into the range where these cells would be effective at effective doses, we hope. And compatible with that, we so far have just tested the function in vitro, and we find that these cells are able to kill BCMA cancer targets very efficiently. So more to do here, but this is highly encouraging that we now have a path towards non-viral single-stranded DNA knock-in to generate high-efficiency T-cell therapies. So now let me move on to the second part. How do we find ways to make CAR T cells or TCR T cells work even better and overcome some of the challenges we know that they'll face? So what are some of the challenges? We know that even if we can make an antigen-specific T cell, if we want to go after major cancers that remain major causes of mortality, solid tumors, we need to make these cells overcome the challenges they'll face. We know that antigen cells, even antigen-specific T cells, even if they get into a tumor, will face a whole number of challenges that limit their ability to actually clear these tumors. They'll face chronic antigen exposure, which can cause dysfunction. They can face checkpoints. They can face suppressive cells like regulatory T cells. They can face suppressive cytokines like adenosine and TGF-beta. So we wanted to see, could we discover genetic edits that we can put into these cells along with the antigen receptor that would make them perform better and clear these tumors more effectively? So this has been the work for several years now of two really talented postdocs. Eric Schiffroot is a postdoc in my lab. Julia Carnevale is an oncologist and a postdoc in Alan Ashworth's lab. They're both about to start their own groups. And they developed an approach to actually do genome-wide screens in primary human T cells. Now, genome-wide screens have been used very effectively in cell lines, in animal models. We had a lot of challenge in developing a system to do this in primary cells until the work of Eric and Julia. What they found was that the problem that we faced initially was how do we get Cas9 in at high enough efficiencies to actually modify a population of cells. This has been done in other systems, either by making transgenic animals or with lentiviral delivery, which was not very efficient in our hands. So what Eric and Julia devised was a hybrid strategy where they can actually infect a population of human primary cells with a pool of guide RNAs in lentiviral format. The lentivirus delivers the guide RNA as well, but we had trouble with the bigger Cas9 to get efficient delivery with lentivirus. So Eric and Julia did a hybrid where after they infect the cells with the library of lentiviral guide RNAs, they electroporate the cells with Cas9 protein. And Cas9 protein will just go in, and we think it just stays around transiently, but that's actually sufficient for Cas9 to find the guide RNA that's expressed off the lentiviral integration cassette and permanently modify the gene target of that Cas9 guide RNA. So what you end up with is a population of human primary cells where each cell has a different genetic edit, and each cell is effectively barcoded by the lentiviral integration cassette, which tells us which gene has been knocked out in each cell. And what we know is that once we have this population of cells where each cell has a different genetic edit, we can race these cells against each other and actually start to learn the lessons of which genetic edits will drive the phenotype that we want the cell to have. And so the first thing that Eric and Julia did was they wanted to see, could we learn the rules of all the genes of the genome that determine the extent to which T cells will proliferate when they're re-stimulated. So in order to do this, what they did was they introduced a library of 80,000 guide RNAs targeting roughly 20,000 genes in the genome to get this genome scale screen. They then labeled all these cells with CFSE, which is a dye that will dilute as the cells divide. And then they did these enormous FACS sorts to separate the dividing cells from the non-dividing cells. They asked which guide RNAs make the cells divide more, or which guide RNAs make the cells divide less well. And what we found is very clear, that if we knock out any of these genes shown here in red, the cells fail to proliferate appropriately when they're re-stimulated. And these are not random genes. These genes actually reconstruct decades of work that have gone into the biochemistry downstream of the Tcell receptor signaling pathway. So this really showed us that the results are meaningful and are pointing to real biology. We were even more excited to find a smaller number of genes, shown here in blue, that when we knocked these out, the cells actually divided more when they're re-stimulated. So we think that these are potentially checkpoints that limit the ability of cells to divide when they're re-stimulated. And that if we targeted them either with drugs or with genetic edits in cell therapies, we could make the cells more potent. So here we've discovered genes that when we knock them out, make the cells divide more. We wanted to ask, do some of them also make these T cells into better cancer killers? And we had to do this in an arrayed format. So we knock out each of these candidate genes one at a time in a different well in antigen-specific T cells. Then we co-culture these antigen-specific T cells, each with a different genetic edit, with cancer target cells that are labeled in red. And we asked which of the genetic edits make the T cells more able to kill the cancer cells. And so if you just put the antigen-specific T cells with the cancer cells, you see that they're able to kill somewhat compared to no T cells. But we actually found 4 genes that when we knock these out, make the T cells much more potent at clearing the cancer. We could actually watch this with live cell imaging dynamically, and we could see that at least 4 genes, when you knock them out, start to clear these tumors better. And at the top of this list is a gene called RASA2. So pay attention to RASA2. That's going to be the theme. So that was doing genome-wide screens with just stimulating the cells. We started to do this initially in the first paper, but now Eric and Julia have amassed a huge amount of now genome-wide screens under multiple suppressive conditions to see, can we find all the genetic edits that will make the T cells proliferate even in challenges that they might face in the solid tumor microenvironment. So they've redone genome-wide screens now in adenosine, in cyclosporin, in tacrolimus. These are immunosuppression drugs. They've done it in the presence of TGF-beta, which is a suppressive cytokine, and co-cultured with regulatory T cells, which are suppressive T cells. They've done these genome-wide screens, separated dividing and non-dividing cells, and learned the lessons. And so we now have hits that will make T cells resistant to each of these suppression conditions, but we started to see that there were actually some genes that were shared. And there were actually 2 genes that were shared among all of the conditions that we tested that continue to make these T cells resistant to these forms of immunosuppression. And that among these 2 genes is RASA2. So RASA2 not only makes the T cells divide more and better cancer killers, it also makes them resistant, now we know, to multiple forms of immunosuppression. So what is RASA2? Well, RASA2 has never really been studied in T cells, and that's the power of doing these unbiased genome-wide screens. But in other cells, RASA2 has been proposed to be a negative regulator of RAS signaling. And we know that RAS signaling does play a key role in regulating T cell responses and feeds into a number of pathways downstream, especially of this T cell receptor and co-stimulatory receptor signaling. This is an old schematic that we've adapted from Anjana Rao and others. So is this in fact what RASA2 is doing in T cells? Is it a negative regulator of RAS? Well, that does seem to be what we're seeing biochemically. We're seeing increased active RAS in the RASA2 knockout cells. But this is really critical. This is only in stimulated cells. So this doesn't appear to be dysregulated RAS signaling. It is potentiated RAS signaling after T cell stimulation. So that is really important, and that seems to be a consistent theme that we're seeing. We're not seeing dysregulated growth. We're only seeing enhanced proliferation after T cell stimulation. And going along with that, we see in these RASA2 knockout cells evidence of increased phospho-ERK and phospho-MEK and also phospho-F6, showing us multiple MAP kinase and mTOR signaling pathways that are enhanced as a result of RASA2 knockout cells following stimulation. Going along with that, we've also looked in reporter lines. Knocking out RASA2 feeds in and shows increased transcriptional activity, especially of AP1 and NF-kappa B, and to a much lesser extent, NF-NFAT. And downstream of this, I'm not going to show you, we've done a lot of RNA-seq. We've really reprogrammed the transcriptional responses of these cells. And an end product of that, for example, is that we see increased cytokine production in stimulated cells that have RASA2 ablation. So that got us thinking that RASA2 really is an important negative regulator of signals. How is this actually found in nature? Well, if we look in existing data sets, in humans, for example, RASA2 will go down in both CD8 and CD4 with acute stimulation. So the cell seems to actually downregulate this break as it responds to stimulation. It doesn't go away completely, but it goes down. But over time with chronic stimulation, RASA2 tends to go up. This is in this is an animal model. This is in human TILs. In the tumors, we can actually see higher levels of RASA2 in T cells than we see in blood, suggesting that RASA2 in chronically stimulated T cells can really be a brake. So can we start to remove these brakes, either for acute stimulation or chronic stimulation? So one thing is that we've seen stronger T cell stimulation responses in the RASA2 knockout cells, but we wanted to see, could we actually quantify this? So we made antigen-specific T cells for NY-ESO and exposed them to different levels of pulsed peptides in antigen-presenting cells, and we actually saw that we're able, that at the lowest doses, these T cells still depend on antigen. They're not antigen independent, but we've made them sensitive to lower doses of antigen, and we've increased the amplitudes of these signals. So does that actually translate to a more effective cell therapy? Well, we think it does. We've actually, in collaboration with Justin Eichwem and a gift from Michelle Sadelain, used cell lines that have CD19, a target of CD19 CAR T cells, expressed at different levels. We know that CAR T cells can fail if the cancer cells have very low levels of the antigen target. So we've actually engineered a system that does this in vitro. And indeed, the control cells fail to kill these cancer targets at the very low antigen density. But RASA2 cells continue to do it. So we don't see much of a difference at the high density, but at low density, RASA2 starts to really distinguish itself. So these cells are resistant to immunosuppression, they're better cancer killers, and they're able to recognize lower antigen densities. Now, one concern we had was if we really ramped these cells up and made them into effective cancer killers that are highly sensitive to stimulation, are they going to burn out? So through chronic antigen exposure, will they start to fail? So Eric and Julia have tested this very systematically. They've created a system similar to what others groups have now published, where we take these antigen-specific T cells and chronically expose them to tumor target cells. So every few days, we will re-expose these cells to more tumors. And indeed, what we see is that over time, the cells will start to fail. But to our delight, the ROSA2 cells do not fail. So even though they're ramped up and they're responding strongly, they continue to respond strongly through multiple antigen exposures. So the control cells will start to fail and the cancer cells will grow out by the last stimulation, after 5 or 6 rounds of stimulation. The ROSA2 cells continue to kill. And this was with a TCR engineered T cell. We've also done this with with these potent CD19 track CARs. And even these potent CARs will start to fail after multiple rounds of stimulation, but the ROSA2 knockout version of this continues to effectively kill the cancer cells in vitro. So that's in vitro. Ultimately, what we care about, will this also work in vivo? And so we've now tested this in multiple in vivo models. So we've started this with the NY-ESO. If we, for example, put NY-ESO specific T cells into a mouse, an immune deficient mouse that has an immune deficient, that has human NY-ESO positive cancer cells implanted in it, the ROSA2 cells do a better job of controlling the tumor volumes than the control cells at this dose that we tested. And we've even done this in the CD19 track CARs. So again, the CD19 track CARs have been published to be among the best CARs that are out there, but we were really astonished to see that the ROSA2 knockout version of the CD19 track CARs do even better. And this translates to really a remarkable improvement in tumor control, shown here with bioluminescent imaging. You can see the control cells, the tumors start to take over these mice, but at this dose, the ROSA2 cells are still keeping the tumor in check, the cancer is in check, and that translates to marked survival even in these good CARs. So that's with a great CAR that's being used, a great CAR against a hematological malignancy. Let me end by saying, does this also boost effects in solid tumor models? And we do see that. This was work that was done at St. Jude's by Giedrich Prince-Ute and Steve Gottschalk, who had also gotten interested in ROSA2. And they have used ROSA2 in an FA2 CAR in a model of pediatric sarcoma, and a model of sarcoma in immune deficient mice. And again, the ROSA2 cells do a better job of controlling this cancer, even in a solid tumor model. So I'll pause there, but let me just summarize that by saying that we've used genome-wide screens to nominate in an unbiased way a hit that puts desirable features into these cell therapies. It makes them resistant to immunosuppression. It makes them sensitive to low antigen doses. It makes them resistant to failure through chronic antigen exposure, and seems to show marked benefit as a module that we can now put in knockout ROSA2, either in TCR engineered T cells or CAR T cells, hopefully for a wide range of indications. So let me summarize what we're really trying to do. We're developing a suite of tools. I've talked about genome-wide knockouts, but we're using complementary technologies to be able to scan through the genome of human immune cells and understand all the sequences that control the critical functions that we care about. As we do that, we'll learn fundamental lessons about how these cells are wired. We'll learn how cells fail in disease states, find new targets for drugs. But we will also find new targets for gene modification and cell therapies. And as we find these critical cell sequences, now we have the ability to actually go into a targeted site of the genome and introduce a targeted rewrite of that sequence using the power of synthetic biology to reprogram the immune cells and give them the properties that we want to precisely make them into the drugs that will be effective for cancer or autoimmune disease or infectious disease, or maybe other types of diseases in the future as we really start to realize their full synthetic powers. And I'll just end by saying that this effort transcends what can happen in any one lab. We are actively building a community through a new and exciting partnership between the Gladstone Institute and UCSF, we have built an institute, the Gladstone Institute of Genomic Immunology, that's weaving together expertise that's already here at UCSF and at the Gladstone, and continuing through rounds of faculty recruitment to build a community that has a collaborative environment to support computational biology, single-cell genomics, functional genomics, CRISPR engineering, synthetic biology, all the way to expertise in clinical cell therapy manufacturing and cell therapy clinical trials. So if you're interested, we hope that we'll have the chance to work with you in the future. And with that, let me thank the people who have actually done this work in my lab and through our network of collaborators, and thank our funding sources, and thank GenScript again, both for the collaboration that we're working on together towards using the single-stranded DNA template as a research tool, and I hope eventually for a therapeutic tool, and thank GenScript for having me today as a speaker.
Thank you very much, Alexander. It was a great talk. We have a couple of questions coming through. So from Jessica, what are the advantages and disadvantages of using this non-viral approach for developing CAR T therapy compared to the traditional lentiviral approach? Do you see this non-viral approach shortening the development time of CAR T for patients, or do you see this approach being applied to personalized CAR T cell therapy development?
It's a great question. I think the answer is yes to both, I hope. Lentiviruses and viral vectors have a long history, so there is an established track record there, and there's some benefit to that. But there is currently a long lead time to make a new clinical grade or GMP viral vector. It's a driver of cost, it's a complicated manufacturing process. It would be great if we can have a chemically synthesized single-stranded DNA or double-stranded DNA, whatever it's going to be, that we can manufacture much faster without the process of going through all the viral packaging steps. So we hope that this will create a much shorter lead time to actually having a cell therapy. We hope that over time this will continue to reduce cost. So we hope it's cost and lead time. But on top of all that, we hope that there's a big benefit of efficacy, and that comes from the ability that the current viral vectors are going into non-targeted sites of the genome. So we have the benefit of having a non-viral system that will be targeted, which we think will give us much more control over the way that we reprogram these cells by going into targeted, hopefully homogeneous cell genome locations to really control the underlying genetic programs in these cells. So it's some combination benefit of all of those things, I hope.
Great. So we're making sure to get down to a bit of material 12 minutes for questions. Question from Claire. Have you tried knocking more than one gene CARs into T cells with this non-viral approach? For example, would it be possible to use two ssDNA templates for encoding two different genes for KI at different sites?
So it's a great question. In our initial paper, we did do some of that. You can knock in things into different locations, and we did see combinations. The efficiency starts getting the double knock-in is some. It's not exactly the two are independent. If a cell has one genetic knock-in, it's actually more likely to get the second, probably because of the state of the cell being competent to undergo homology-directed repair. So there's some challenges in efficiency, but there is actually a way that those events are not independent. Now, the other challenge is that every time you introduce another knock-in event, you're introducing another cut in the genome and probably going to introduce translocations and other complicated DNA repair. That's not necessarily a block to doing it, but it's something that needs to be considered. An additional approach that we have pushed hard on is actually engineering polycistronic sequences. So I showed a little bit of this. The TCR sequence that we actually have has P2A elements, these peptide fission sequences that separate multiple components, so the TCR alpha and beta, for example. We've also found that we can actually add an extra 2A element, so you can have TCR alpha, beta, plus an extra gene that boosts the function of those cells potentially. So we can actually start introducing on one knock-in construct a complicated polygenic knock-in construct.
Do you sort edited T cells to get pure populations? what systems are you doing to expand the primary cells after electroporation?
Sorry, I'm getting some comments here that I'm muted. Are you able to hear me?
I can hear you, Alex.
Okay, great. Could you ask the question again?
Yeah. Do you sort edited T cells to get pure populations? Which systems are you using to expand the primary cells after electroporation?
Yeah. So for different applications, we either enrich the edited cells or don't. Sometimes the cells actually enrich themselves. So, for example, you could enrich the CAR T cell positives, but you might not need to. The other ones might be neutral. And the expansion protocols that we've used have varied. We try to model it after expansion protocols that have been used in CAR T cell therapies currently, trying to take advantage of cytokine conditions, stimulation conditions that have been well worked out over time. There's continued room to optimize some of that. I think it's generally compatible. What we find is that after electroporation, there's some drop-off. The electroporation is harsh on cells. So maybe something like somewhere between 40 and 60% of the cells will die post-electroporation. But the cells that survive and are knocked in, they tend to expand with the same kinetics as the non-knock-in cells. And so we're able to really expand these cells and get large numbers of knock-in cells within days. And the toxicity has continued to fall as we modify these protocols. So I think that there's an opportunity to get a huge number of cells if you want, and the T cells expand really well. The electroporation toxicity is one thing. We've really reduced the toxicity that comes from the other knock-in components, the Cas9 RMP and the DNA repair template.
Thank you. From Kosovo. How long is the longest ssDNA donor template you've used, and what is the ssDNA synthesis scale you would need for a single patient?
So we are continuing to push the limits of the payload size. I don't know if we've hit an absolute limit yet. We find that there's some trade-off. The efficiency of knock-in goes down with bigger and bigger knock-in templates. We have knocked in things mostly with double-stranded DNA at bigger sizes, several kb, and are starting to convert those to single-stranded through conversion methods in-house. The question for how long a single-stranded DNA can be chemically synthesized is really a question for GenScript, and we're continuing to work with them to see how long a chemically synthesized DNA template can be and what the scale of manufacturing would be. So far, as I showed, it does, at least at the size required for a CAR knock-in, it does seem that we're able to generate enough single-stranded DNA material to generate enough CAR T cells that would be compatible with an eventual cell therapy.
Thank you so much. Question from Sammy. How would you compare transposon-based integrations with site-directed CRISPR integrations in terms of off-target effects, efficiency, etc.?
Yeah, there's been a lot of interest in transposase-based methods. There's been interest because they have potentially huge DNA payloads. They are less targeted than true homology-directed repair. There have been some efforts to try to direct where the transposons go to some extent, but I think the scarless knock-ins that are capable with homology-directed repair still have an edge on specificity. So there's some trade-off between potentially from payload size and other things. There's been some reports of malignancy with transposase-based CAR T cell therapies, and the mechanism of that, to my knowledge, is not still fully clear, but needs to be carefully looked at.
Okay, another question from Subham. Does disrupting cell cycle checkpoints of T cells affect the possibility of autoimmune reactions?
Yeah, so this is a really important question. So there's two things that we're really want to pay attention to. As we juice up these T cells to make them more potent, we want to make sure that we're not transforming them into malignancy. And that's why I emphasize so much, especially if we're going after a RAS signaling pathway. This is a known potential oncogenic pathway. we want to make sure that these cells are not going to have dysregulated growth and become T-cell malignancies. Now, so far, that is not what we have seen. These cells, the ROS2 knockout cells, continue to be regulated, and their proliferation seems to really only be enhanced after T-cell receptor stimulation. And we've done some experiments starting to look even in vivo, and does seem to be dependent on antigen. And with those experiments, we're continuing to work on more data. I'm actually not sure the latest in the lab right now. We're working on that actively for a safety profile there. The other thing is, if we juice up the sensitivity of these cells, will they potentially also cause autoimmune disease? There is the potential, and actually there's been some, our collaborators have noticed that there might be, or that there seems to be more graft versus host disease if the CAR is not a strong CAR. Now, we have gotten around this, and we've not seen this because we're doing the track CARs. So as we manufacture the CARs, we're actually getting rid of the cell's own T-cell receptor. So the potential for graft versus host disease is actually drastically reduced. We're actually getting rid of the cell's own T-cell receptors. They won't be recognizing antigens through that mechanism. They really will only be recognizing antigens through the CAR that we've put in. So I think that might be an important way to do it if we're going to make cells that are much more sensitive to antigen stimulation to prevent autoreactive cells.
Interesting, thank you. We can take 2 or 3 more questions. From John, have you tried to use the ssDNA knock-in method in mouse T cells as well? Do you see a similar improvement in knock-in efficiency without loss of viability?
We've tried, I don't know how much we've tried the single-stranded data. We've tried a lot of things that work well in human cells in the mouse, and it has been an ongoing challenge. I think that we hope that there will be some workaround for this in the future. This has been a major challenge of taking these non-viral systems and taking it to mouse. Now, I don't know what that is. I don't know if there's some, a lot of these steps have just been challenging to move over to mouse, whether it's the culture steps or the electroporation sensitivity or the repair machinery. It's hard for me to exactly isolate what the difference is, or maybe there's multiple, but our efficiencies, we've done it somewhat in mouse, but our efficiencies have really been limiting in mouse. And so we have not been able to really succeed in doing the things that we can do in human cells in the primary mouse cells. If anyone knows why, we're very curious.
Thank you, Alexander. What is the length of the truncated Cas9 binding site on each end in your design?
I would refer people to the Nature Biotech paper, where we've actually carefully done a titration and looked at different truncations and shown exactly the effect of those different truncations. So that is all included in that Nature Biotechnology paper.
Question from James. CAR T cells are often rejected or not received well. Have you looked into CAR MAC or CAR NK cells? I am aware that there are some clinical trials for CAR NK cells to treat solid tumors.
Yeah, I'm not sure about the rejection part and what exactly was being referred to. I guess one thought maybe that this is potentially looking at, there's been discussion of could NK cells be used in another donor, and maybe that's what the question is about the potential to use these in so-called allogeneic treatments, where you have an off-the-shelf or a good donor that then donates these cells that get used in another patient. Now that's a really exciting development, and that has recently been done with NK cells, where there's been cord blood that has been used actually to generate CAR T cells or CAR NK cells. What the effectiveness still needs to be determined over time, but the idea of being able to move into cell populations that can be used in this allogeneic or off-the-shelf way is a major goal of the field. If we could do that, then we could generate big pools of these cells and really start to have something that looks a lot more like a conventional drug, where it's manufactured in the central facility and then dosed out in ways that it would be more consistent than the patient-specific. I think there's 2 routes. Either we have to make the patient-specific much more efficient, so like something that could happen at the bedside or very low doses or even in vivo, or we'll have to start thinking about allogeneic, where we can do these kinds of off-the-shelf approach. I think for different diseases, we might want different things. I think both of those really need to be explored carefully, and I hope that the combination of the non-viral manufacturing with either better delivery for patient-specific or a capacity to do allo, will really continue to move this field forward and expand the reach of these therapies to more disease indications and make these treatments more accessible and more affordable.
Thank you, Alexander. That's all we have time for. Thanks, everyone, for submitting your questions, and we'll see you at the next presentations, which are about to start shortly.
Thank you so much.